Adeno-Associated viruses, or AAVs, were discovered more than 50 years ago. At first, they were not interesting to researchers because they were not associated with any diseases or infections. However, they are now one of the most promising tools for potential gene therapy applications. In the first 20 years, research was focused on the basic elements of AAV genome structure, growth cycle, and latency, culminating in DNA sequencing in the 1980s. During the next 10 years, important developments including experiments revealing the unique molecular functions and features of AAV, refining the basic AAV vector design, and early in vivo experiments and clinical trials. Since then, AAV vectors have been recognized as an attractive mechanism for gene therapy due to their ability to integrate into the genomes of both non-dividing and dividing cells, their specificity, and their minimal pathogenicity.
The general genome structure of AAVs is a single-stranded linear DNA consisting of about 4,700 bases. AAV genomes consist of groups of genes flanked by inverted terminal repeats and a poly-A tail. The inverted terminal repeats are located at each end of the genome sequence, and they consist of an average of 145 GC-rich base pairs, which create T-shaped hairpin structures. These structures serve as primers to facilitate viral replication and integration. AAVs have highly conserved genome groups. The rep group codes for rep proteins, which assist in replication. Key rep proteins are regulated by the P5 and P19 promoters and are produced via alternative splicing. They include rep 78, 68, 52, and 40. Meanwhile, the cap group encodes for viral capsid proteins VP1, 2, and 3. Adenoviruses themselves consist of that linear genomic sequence and the protein shell or capsid that encapsulates it. Within the capsid, there are pre-packaged integrase and protease enzymes.
After entering a host cell, AAVs enter a lytic stage in order to replicate. This requires the presence of a helper virus, such as an adenovirus or a herpes virus. AAV borrows proteins such as DNA polymerase and helicase for viral genome replication, genome expression, and virion production, enabling viral proliferation via the destruction of the host cell. But the greatest therapeutic asset of the AAV vector is its ability to enter a lysogenic replication cycle. After the AAV enters the cell via endocytosis, the viral genome, or the recombinant genome of interest in gene therapy applications, is preferentially integrated into a specific site in human DNA. This site specificity is mediated by the aforementioned rep protein, which has a binding site on the long arm of human chromosome 19.
The advantages of AAV become salient in contrasting AAV vectors with other viral vectors used in gene therapy. Adenoviruses cannot enter a lysogenic cycle, and while retroviruses may do so, their integration into the genome is highly unpredictable. There are some disadvantages to using AAV for gene therapy. Vectors can be very difficult to produce and can only carry a small amount of DNA. AAV can only carry about 4 kilobase pairs of DNA, while adenoviruses and retroviruses can accommodate about double that. However, recent research has made strides in increasing the packaging capacity of AAV. One strategy in development is the splitting of transgenes into smaller plasmids. Ultimately, there are several exciting possible uses of AAV that could someday be applied in a clinical setting. It may be used to increase neuronal activity of Parkinson's disease patients, and possibly even reverse its effects. A 2018 review by Tobias M. Axelson and David P. D. Woldby explains how the use of AAVs to deliver DNA coding for a variety of neurotrophic factors, which are proteins that support neurons' growth, survival, and differentiation, may continue to improve the efficacy of gene therapy for Parkinson's disease. Still, other applications of AAV-mediated gene therapy, like the reversal of intervertebral disc generation, are currently in the clinical trial phase of development.